Hi guys, in this video I am going to talk about Sanger sequencing. Sanger sequencing is the process of selective incorporations of chain terminating dideoxynucleotides by DNA polymerase during in vitro DNA replication. The sequencing is known as Sanger sequencing because it was discovered by Frederick Sanger. The another name of Sanger sequencing is Dideoxy sequencing because we used dideoxyribonucleotide to stop or terminate the polymerizations of DNA in vitro condition. So, what is dideoxyribonucleotides or DDNTP? At first, we should know the structure of ribonucleotide, deoxyribonucleotide and dideoxyribonucleotide. Now I am drawing ribonucleotides or NTP. This is 1 number position where base and hydrogen present. This is 2 number position, this is 3 number position, this is 4 and this is 5 number position where the phosphate groups are present. Now I am numbering. Here 2 or 3 number position OH groups are present. Now I am drawing deoxyribonucleotides or DNTP. Now I am numbering 1, 2, 3, 4, 5 deoxyribonucleotides or DNTP. Here in the 2 number position the OH group is absent and I am now drawing dideoxyribonucleotide. This is 1, 2, 3, 4, 5 and in 2 and 3 their OH group are absent. They are replaced by hydrogen atom. For nucleotide polymerization we need template, DNA strand, a primer which gives us free 3' OH group which are required for DNA polymerase at the DNA nucleotide position. This is the template and this is the 3' OH group present in primer and this is the DNA polymerase. Now let's suppose new DNTP or deoxyribonucleotides come and this 3' OH or hydroxide groups are present in the 3' position interact with 5' phosphate group. This upper the phosphate group will remove and they create a phosphodiester bond in 3' OH of 5' phosphate group, and in this way the polymerizations can be done. And I already told that DNA polymerase requires a 3' OH hydroxyl group present in the 3' position of a nucleotide group to make a phosphodiester bond to form nucleotide. It means it is very essential for DNA polymerization. Now, when dideoxyribonucleotide or DDNTP come, it also here 3' OH interact with 5' phosphate group. This 2 phosphate group remove as phosphate they create a phosphodiester bond, and they create a bond in between 3' OH and 5' phosphate group. But here, after the addition of DDNTP, in this position the further new nucleotide cannot be attached, as there no OH group is present. As we remember the structure of DDNTP, we can understand that there no OH group is present in their 3' or 3 number position. As we compare with NTP or DDNTP, they are present OH group in their 3 number position, but in DDNTP there are no OH group or functional group which is required for DNA polymerase reaction or in DNA polymerization. In the presence of DDNTP or dideoxyribonucleotide, it may be ATP, GTP, CTP or DDTTP, polymerization will stop because in the DDNTP has no OH functional group in the 3' position of primer. Here primer is essential for DNA synthesis. Primer provides initial 3' hydroxyl group to form a phosphodiester bond with incoming DNTP or deoxyribonucleotide. When DDNTP is added in a DNA synthesis reaction terminate with the incorporation of DDNTP or dideoxyribonucleotide. Here, so this reaction is also called chain terminating method. This is also called chain terminating method. And here DDNTP are also known as chain terminating nucleotides. And this is the principle of the Sanger sequencing or chain termination method or dideoxy sequencing. Now I am going to talk about the procedure. This is a double stranded DNA which I am drawing now. This is the double stranded DNA and this is the template. And we want to sequence the strand. Now we are denaturating the double stranded DNA and we will get single stranded DNA, and write the single stranded DNA, which we want to know the order of nucleotide which are present in the single stranded DNA. We used it for the template for the nucleotide polymerization. At first, we require a radio level primer which attached to the template DNA at the end of the complementary base pair. And after that, polymerizations will be done by DNA polymerase. And here radio level is required for the detections of DNA after gel electrophoresis by autoradiography. Radio labeling is required to detect the newly synthesized DNA after gel electrophoresis. Now the process is very important, which is chain terminating or chain termination reaction. We have to all components for the polymerization into these 4 reaction mixture tube. The common reaction components are many copies of DNA template stand, many copies of radiolabeled DNA primer, DNA polymerase, and deoxyribonucleotides, maybe DATP, DCTP, DGTP, DTTP, large amount of deoxyribonucleotides, all are the common reaction components. Now we have to add only one dideoxyribonucleotides in each reaction mixture tube to stop the polymerization. So in one mixture tube we add DDATP in 2, DDCTP in 3, DDGTP, and in 4 DDTTP. So GNTPs reacts with DDNTPs and we will get fragments from here. And let's see what happens. At first, for the case 1, we write the primers and we write the stand which we want to sequence this stand. And in the mixture tube 1, we add DDATP as dideoxyribonucleotides. We know that adenine conjugates with thymidine and guanine conjugates with cytosine. Please remember that, and this adenine terminate the thymidine position. So we get from here 3 fragments. One is 5'A because it terminates thymidine. So we, as our sequence stand, we will get 3 fragments from here. One is 5'A, one is 5'ATGCA, and next one is, which is 5 nucleotide is present, and next one is 10 nucleotides is present. And for the case 2, at first in every sequence we have to write the sequence stand, we have to write the stand which we want to sequence, and in the case 2 we add DDCTP as dideoxyribonucleotide, and we know that cytosine conjugate guanine. So we get here from this stand where the guanine is present, the cytosine terminate, and we will get fragments. And from this stand, by adding DDCTP, we will get 4 nucleotide fragments, 6 nucleotide fragments, and 12 nucleotide fragments. This is the size of the fragments. And for the case 3 or mixture tube 3, our DNA stand is TACGTGAACTAG 3' to 5'. Here we add DDGTP, and we should remember guanine conjugates with cytosine. So here this DDGTP break or terminate at the position where the present cytosine is present. So we will get from here 3 nucleotide fragments and 9 nucleotide fragments. For the case 4, we add here as dideoxyribonucleotide DDTTP, and we know that thymidines conjugate with adenine. So here present adenine will terminate by adding this DDTTP, and we will get here 4 nucleotide fragments where the adenines are present, and those are 5'80 where 2 nucleotides present, 5'80GCACT 3' where 7 nucleotide present, here 8 nucleotide present, and here 11 nucleotide present. Please follow our instruction. Now, getting the fragments, we have to separate the DNA fragments according to their size by gel electrophoresis. But before run, in gel, we have to denature this newly synthesized fragment from the template stand, then we have to run in the gel electrophoresis. Now in gel electrophoresis, DNA fragments are present in different length. The presence of nucleotides in the fragments, it shows its length or size. Please remember the sequence and we will get this type of bands. And another way or which we require, which we want to sequence the stand, we will get from here. Please notice carefully. Now I am write the original DNA stand, which were 5'GATCAAAGT GCAT 3', and this is the original template. And G break the G conjugate with cytosine, C conjugate with guanine, T conjugate with adenine, and adenine conjugate with thymidine. This is the basic concept to get a sequence from a stand. So I write the original template, which is 3' TACG TG AAC TAG 5', and the sequence of this stand, which we get from this by using Sanger sequencing, that is 5'ATGC ACTTG ATC. If we see that, the basic concept is guanine conjugate with cytosines and adenine conjugate with thymidine. Hope this video will help you to understand the Sanger sequencing.